Histology is the study of tissues and their organization within the body. It is a crucial field of study for medical students, as it provides a foundation for understanding the structure and function of various organs and systems. One of the key types of tissues we focus on in histology is epithelial tissue. In this script, we will explore the different aspects of epithelial tissues, including their classification, cellular junctions, specialized structures, and their role in different organs and systems. Epithelial tissues are one of the 4 primary types of tissues, along with connective, muscular, and nervous tissues. They cover the surfaces of organs and line the cavities within the body. Epithelial tissues are primarily involved in protection, absorption, secretion, and filtration. These tissues are characterized by their tightly packed cells, with little to no extracellular matrix. The cells are held together by specialized junctions, which we will discuss further in the next section. Epithelial cell junctions play a crucial role in maintaining tissue integrity and function. There are 3 main types of cell junctions in epithelial tissues: tight junctions, adherens junctions, and desmosomes. Tight junctions form a barrier between adjacent cells, preventing the movement of substances between them. Adherens junctions provide mechanical support and act as a belt-like structure to hold cells together. Desmosomes are spot-like structures that anchor cells together, providing strength and resistance to mechanical stress. Epithelial cells display various apical specializations that reflect their specific functions. Microvilli are finger-like projections found on the apical surface of absorptive cells, increasing their surface area for absorption. Stereocilia, on the other hand, are long, immotile microvilli found in specific epithelial cells, such as in the epididymis and inner ear. Cilia are motile structures and are found in respiratory epithelium and the female reproductive tract. These structures play important roles in facilitating the movement of substances across the epithelial surface. Beneath the epithelial cells, there is a layer known as the basal lamina, which is a specialized extracellular matrix secreted by the epithelial cells themselves. The basal lamina provides structural support and acts as a selective barrier between epithelial cells and underlying tissues. It also participates in cell signaling and differentiation processes. The basal lamina, together with an underlying layer of connective tissue, forms the basement membrane, which supports and anchors the epithelial cells. Glandular epithelium is a specialized type of epithelium that is responsible for secretion. Glands can be classified as endocrine or exocrine based on their mode of secretion. Endocrine glands secrete hormones directly into the bloodstream, while exocrine glands secrete their products through ducts onto epithelial surfaces. Exocrine glands can further be categorized into merocrine, apocrine, and holocrine glands based on the nature of secretion. Squamous epithelia are characterized by thin, flat cells with a flattened nucleus. They line structures such as blood vessels, serous membranes, and air sacs in the lungs. Squamous epithelia provide a barrier against friction, control the diffusion of gases and fluids, and participate in filtration processes. Columnar epithelia consist of tall, narrow cells with elongated nuclei, often appearing as a single layer. They line the digestive tract, where they play a role in absorption and secretion. In areas such as the small intestine, columnar epithelial cells possess microvilli on their apical surface, further enhancing their absorptive capacity. Cuboidal epithelia are characterized by cube-shaped cells with a round or centrally located nucleus. They line structures such as kidney tubules and glandular ducts. Cuboidal epithelial cells are involved in secretion, absorption, and the formation of barrier functions. Neuroepithelium refers to the specialized epithelial tissue found in the sensory organs, such as the retina of the eye and the olfactory epithelium. These epithelial tissues are involved in the perception and transduction of sensory stimuli, converting them into electrical signals that can be interpreted by the nervous system. Urothelium, also known as transitional epithelium, is a unique epithelial tissue found in the urinary system. It lines structures such as the urinary bladder, ureters, and part of the urethra. Urothelium is specialized to withstand stretching and changes in volume, as it undergoes reversible changes in cell shape and organization. Epithelial tissues can undergo pathological changes, including metaplasia, dysplasia, and neoplasia. Metaplasia refers to the reversible transformation of one type of epithelium into another due to chronic irritation or inflammation. Dysplasia, on the other hand, refers to the abnormal growth and organization of epithelial cells, which can be a precursor to cancer. Neoplasia is the formation of new, uncontrolled growth of epithelial cells, leading to the development of tumors. Epithelial tissues interact closely with the surrounding mesenchyme, which is a type of connective tissue. These interactions are critical for tissue development, repair, and regeneration. Epithelial mesenchymal interactions involve signaling molecules and physical cues that regulate cell differentiation, migration, and tissue morphogenesis. In conclusion, understanding the intricacies of epithelial tissues is essential for grasping the underlying principles of organ structure and function. Epithelial tissues exhibit a remarkable diversity, both in their cellular organization and specialized functions. By comprehending the different types of epithelial tissues, their cellular junctions, and specialized structures, medical students can gain valuable insights into the role of epithelial tissues in maintaining homeostasis and the development of diseases.